Hi all, welcome to Physio Exam Buddy. This video is on the auditory pathway as well as deafness. One of the recently asked long essay question for physiology exam is draw and label auditory pathway showing receptors and different synapses. And the sub question was describe the different types of deafness with the tests used to differentiate them. So in this video deals with the auditory pathway as well as the different types of deafness. Like any other pathway, the auditory pathway also starts from the receptors. So here the auditory receptors are mainly the hair cells which are present in the organ of cortex. So this is the first diagram you are supposed to draw in auditory pathway, that is the structure of the organ of cortex, and you have to draw the outer hair cells as well as the inner hair cell which are the auditory receptors. And the cell bodies of the sensory neurons, they arborize around the bases of these hair cells and they forms the spiral ganglion. And from the spiral ganglion, the axons of the afferent neurons innervate the hair cells forms the auditory or cochlear division of the eighth cranial nerve. You know, the eighth cranial nerve is the vestibulocochlear nerve and it is mainly concerned with both balance as well as hearing. That is, the cochlear division is the one which is mainly related to the hearing. So from the auditory receptors, the cell bodies, they forms the spiral ganglion and the axons, they forms the cochlear division of the eighth cranial nerve. And from the eighth cranial nerve, the fibers will enter the dorsal as well as the ventral cochlear nuclei, which is situated in the medulla. And from the medulla, the fibers moves into the pons, that is into the superior olivary nucleus in the pons. Most of the fibers actually go into the contralateral superior olivary nucleus, whereas some fibers, only few fibers will move into the ipsilateral superior olivary nucleus. And from the superior olivary nucleus, the fibers moves into the nucleus of the lateral lemniscus, which is again present in the pons. For the easy understanding or remembering the auditory pathway, you should know a mnemonic, which is the E. coli MA. E is the eighth cranial nerve and C means the cochlear nuclei, that is dorsal as well as ventral cochlear nuclei. Now you know the fibers goes into the superior olivary nucleus. So this O is for the olivary nucleus. Then L for the lateral lemniscus, that is eighth cranial nerve. Then fibers move to the cochlear nuclei, then O for olivary nucleus and L for the lateral lemniscus. And from the lateral lemniscus, the fibers goes into the inferior colliculus of the midbrain. And from the midbrain, the inferior colliculus, the fibers goes into the medial geniculate nucleus of the thalamus, and from there arises the auditory radiations which finally end in the primary auditory cortex. So the continuation of the mnemonic: E is eighth cranial nerve, C cochlear nuclei, O superior olivary nucleus, L lateral lemniscus, then I is our inferior colliculus of the midbrain, and M is the medial geniculate body, then A is the auditory radiations or the auditory cortex. Now we will study in detail about the auditory pathway. So this is the diagram you are supposed to draw when auditory pathway is asked, and it is from the guidance. So this pathway starts from the auditory receptors, which are the hair cells in the organ of cortex, whose cell bodies give rise to the spiral ganglion, and from the axons arises the cochlear division of the eighth cranial nerve. And this eighth cranial nerve will finally terminate in the medulla, that too in the dorsal as well as in the ventral cochlear nuclei. So till here, till the dorsal as well as ventral cochlear nuclei, these fibers, they forms the first order neurons. And from the first order neurons, that is from the cochlear nuclei, arises the second order neurons. We will see the second order neurons, that is from the cochlear nuclei to the contralateral superior olivary nucleus, mainly the fibers goes, whereas some or few fibers goes to the ipsilateral superior olivary nucleus in the pons. And you can see here that in the medulla, these crossing fibers, they form a conspicuous mass which is known as the trapezoid body. So up to the superior olivary nucleus in the pons, the fibers belongs to the second order neurons, that is from cochlear nuclei to superior olivary nucleus forms second order neurons. And from the superior olivary nucleus, the fibers ascend as the lateral lemniscus, whereas some of the cochlear fibers, they again go back to the nucleus of the trapezoid body and from there back again into the lateral lemniscus. And some of the cochlear fibers also can goes directly to the nucleus of the lateral lemniscus, and from the lateral lemniscus the fibers goes into the inferior colliculus of the midbrain. So these neurons, that is from the superior olivary nucleus to the up to the inferior colliculus, they belongs to the third order neurons. And from the inferior colliculus, the fibers goes into the medial geniculate body and they forms the auditory radiations and finally ends in the primary auditory cortex, which is the area number 41 or the superior temporal gyrus. And in the auditory cortex, the low tones are represented basically in the anterolateral portion of the primary auditory cortex, whereas higher tones, they got their representation on the posteromedial portion of the primary auditory cortex. Along with the primary auditory cortex, which is represented via a blue color, there are certain association areas, that is the auditory association area surrounding the primary auditory cortex, which also helps in the hearing mechanism. So first association area is the Brodmann's area number 22, and it helps to process the auditory signals mainly related to our speech, and they are more active on the left side, whereas on the right side it is concerned with the melody, pitch as well as sound intensity. And the next association area is the planum temporale, which is a portion of the posterior superior temporal gyrus, and it is again larger on the left side in right-handed individuals. And the asymmetry is more common in case of musicians, and they are mainly concerned with the language-related auditory processing. Now we will see some of the special features of the auditory pathway when compared to other sensory pathways. One of the most important feature of the auditory pathway is that the spatial orientation is present. And each auditory cortex will receive the input from both sides. That is, if in the primary auditory cortex of the left side will receive input both from the left as well as the right ear. And also the signals from both the ears are transmitted through pathways on both sides. And you can see here the crossing over occurs at three different regions in the auditory pathway. First one is at the level of the trapezoid body, and the second one is at the level of the commissure of props, which is at the nucleus of the lateral lemniscus. And the crossing, it is not shown here, but there is a crossing here, that is at the level of the inferior colliculus. You can actually draw this crossing that at the level of inferior colliculus, that is the third crossing of the auditory pathway. Next special features are the collateral fibers from the auditory pathway pass to the reticular formation. And another special feature is that it will project diffusely into the and causes the activation of the entire nervous system in response to the loud sound. The fibers are also sending collaterals to the cerebellar vermis. The fibers also from the trapezoid nuclei, it moves through the medial longitudinal fasciculus to the cranial nerve nuclei also. And the fibers from the nucleus of the lateral lemniscus will also found to be going into the superior colliculus. So these are some of the special features of the auditory pathway. Now we are moving on to the second question, that is the sub question of the long essay, that is deafness. Please remember that the central lesions in the auditory pathway does not produce unilateral deafness. They mainly interfere with the sound localization as well as discrimination, whereas unilateral lesions of the cochlear nucleus as well as other peripheral structures will only result in unilateral deafness. So we will see in detail about the deafness. Deafness means either reduced or absent hearing. There are two types of deafness, that is conduction deafness is also there, and again nerve deafness is there. Conduction deafness means either occurs impaired sound transmission from the internal from the external or the middle ear. So any impaired sound transmission from the external and middle ear result in conductive deafness. And the causes for conduction deafness are anything that impairs the conduction of the sound, like plugging of the external auditory canal with wax, or destruction or fibrosis of the ossicles, then thickening or rupture of the tympanic membrane, or also otosclerosis. Next is about nerve deafness or sensorineural deafness, and it is mainly due to the damage to the hair cells or the neural pathways. And the main causes for sensorineural or nerve deafness are drugs like aminoglycosides, then prolonged exposure to loud noise, then tumors of eighth cranial nerve, or any tumors at the cerebellopontine ankle, then vascular damage occurring in the medulla, and also presbyacusis are some of the causes for the sensorineural as well as nerve deafness. Next, about tests to differentiate the deafness. The main test is the tuning fork test, which we are usually employed in our practical labs. So the tuning fork test include Rennie's test, Weber's test, Schwabach's test, as well as ABC. And how these tests you are doing, everything you are actually studying in the practical. So I am not going to explain it again. Please remember that normally in case of Rennie's test, the air conduction is better than bone conduction. That is how you are writing the interpretation. But in case of conductive deafness, you can see here that the bone conduction will be better than that of the air conduction. Whereas in sensorineural or nerve deafness, again the air conduction will be better than bone conduction. Next is the Weber's test. Weber's test is normally central, or it is not lateralized in normal individuals. Whereas in conductive deafness, the lateralization goes to the poorer ear. Whereas in sensorineural deafness, it is lateralized to the better ear. In Schwabach's test as well as the ABC test, that is absolute bone conduction test, both conduction is same as that of the examiners in case of normal individuals. Whereas in conductive deafness, the bone conduction will be same or better as that of the examiners in both Schwabach's as well as in the absolute bone conduction test in conductive deafness. And in case of nerve deafness or sensorineural deafness, again, both the tests, the bone conduction is found to be reduced. And other test which is used to measure the degree of deafness to various frequencies is by doing an audiometry, and the record obtained by doing the audiometry, it is known as the audiogram. So we are actually giving pure tones of different frequencies, and they are presented through the earphone, and the threshold intensity of each frequency is determined, and it is plotted on a graph as percentage of normal. So this is a typical audiogram which you get in case of sensorineural or nerve deafness. That is, in the y-axis you will get the sound intensity, and in the x-axis you will get the frequency. So you can see here that in nerve deafness, you know that both the air conduction as well as the bone conduction will be decreased. So here the X mark, that is the cross mark, shows the air conduction as well as the star mark represents the bone conduction. So both these will be decreased in case of nerve deafness or the sensorineural deafness. Whereas you can see here in conductive deafness, the bone conduction is normal. That is star. This is normal. But the air conduction, that is the ossicular conduction, is decreased at all frequencies. So there would be a large air-bone gap. If the air-bone gap goes above 10 Hz, then it is actually deafness, and it mainly indicates conductive deafness. So this is the audiogram findings of the conductive as well as the sensorineural deafness. And other types of audiometry used in clinical practice are impedance audiometry, Bexy audiometry, as well as brainstem evoked response audiometry. So that is about the auditory pathway, different types of deafness, and the different tests to differentiate the two types of deafness, that is conductive deafness as well as sensorineural deafness. Thank you.